It's a question that leaves the minds of those in the medical community spinning. Why do medications that help the vast majority of patients sometimes cause violent reactions in just a few people? After all, new medications are exhaustively studied.
We understand the biology of the disease. We understand what targets we want the drug to interact with. We understand sometimes hitting one target is not enough because of redundancy of mechanisms. You want to hit more than one target. And we understand in some cases that different patients respond differently.
So, to answer that why question, pharmaceutical companies call on the scientists at the Hamner UNC Institute for Drug Safety Science. It's one of only 2 such research facilities in the world.
We have the ability to explain why, and that's helpful in informing the next drug to be made to make it more safe. But very recently, we've been able to go farther than that and actually not just explain why, but figure out a strategy to control or manage that risk, usually personalized medicine. So we now believe we can use the tools that we've created over the last 6 years to solve the problems by being able to identify the rare patients who are going to have these severe reactions to the drugs.
Medicines work the way they are supposed to on about 99% of patients. The odds of a patient experiencing a rare drug reaction are remote, maybe 1 in 10,000. But if it happens, approval of a new drug can be delayed. That not only lengthens the amount of time it takes to get a drug to a patient, it's also expensive. Consider, it takes almost 10 years and almost $2 billion to bring a new drug from discovery in the lab to when it's sold on a store shelf. So, the Hamner Institute, with its history of studying chemical safety, partnered with the University of North Carolina at Chapel Hill Schools of Medicine and Pharmacy to better understand what are called rare drug reactions.
So in the blue, we're looking at the nucleus of the cell, and showing in the red, we're looking at the mitochondria activity of the cell. We're showing here, we're looking at cytochrome C, is a cyan type color here.
Joe Trask uses cellular imaging to study a cell's health. Individual cells are a lot like the people they make up. You can learn a lot from a snapshot.
So we're interested to determine if cells are undergoing some programmed cell death pathway. Has the membrane been compromised? Has there been any kind of perturbations to the cell? We're painting or staining the cells with fluorescent probes, and these probes will identify the nucleus of the cell so we can determine the size, shape, and also being able to identify where the cell is actually in the image so we can actually make measurements on it.
There's something genetically different about that group of people that's making them susceptible to the toxicity.
Dr. Mary Mosdale believes part of the answer to a drug's toxicity can be found in the patient's genes.
And if we could identify the particular gene variants that are causing the drugs to be harmful for that small group of people, we can screen people and identify those people that are going to have a positive response with no harm and people that are going to have a harmful response, or they'll be susceptible to the toxicity, and keep just those people from taking the drug and let everyone else take the drug safely.
Scientists are also finding there are many reasons for drug reactions. That's because people are complicated. There are environmental factors, aging, multiple illnesses occurring at the same time. Researchers now believe combining genetic tests with protein markers in the blood and urine can answer the why question.
And that's really what we do here is focus the science and the techniques that are currently available, not to see who will have the greatest benefit, but to identify the rare patient that will have harm. And I think the most exciting thing to us is that we can get drugs with this technology to the patients quicker and at much lower cost, which will translate into cheaper medications.